like to welcome Dr. Fenton. The next presentation.
Well, good morning, everyone. It's wonderful to be with you in person, and I'm going to describe my work in schizophrenia, which, truth be told, is I don't work on schizophrenia. In fact, I don't work on any neuropsychiatric diseases in particular, but I work on how to think about them. I'll give you an example of one of the things that we've done when I was at Downstate. We took the drug PCP, angel dust, and cyclidine, which is known to induce psychotic-like symptoms in people and also in rodents. And we gave that drug to rats, and we wanted to be able to, measure what the consequences were for the rats' thinking, if you will. And so the best way for us to think about how to do that was to take a cognitive signal that we can extract from the rodent brain, and that's the place cell signal. And so what you're seeing here are the firing rate maps. Firing rate maps, can you see a cursor? Well, in any case, those are firing rate maps. The first row of them are before the injection, and these are hippocampus CA1 cells recorded while the animal is walking around the environment. And you can see that each cell is discharging in a particular region of the space, and that tuning, if you will, to location is called the firing field. And you can imagine from those data how the animal might make a map, if you will, of its environment, how it might think about where it happens to be as it walks around in space. On the bottom row is after the PCP dose that we gave, and you can see that there's very little change. And so that was quite puzzling. And in fact, it turns out a number of colleagues have made the same measurements and not reported them because it's not that remarkable. But if you actually ask the animal to do a task after being given this drug, the animals are impaired. And you can see here a video on the left is after a control dose. An animal is very capable of avoiding being in that area that's marked red. And on the right side, you can see sometimes it's able to avoid, and then it'll make a series of mistakes. And you can quantify that if you look in the at the bars up here. And the thing to recognize here is that the animal has to ignore where it happens to be on the floor. The arena is rotating. There's stimuli on the floor that the animal can smell, and it has to ignore the locations of the shock when it encounters them on particular parts of the floor, and pay attention to where it happens to be in the room. And we think of that as a cognitive control challenge, which is an impairment you can recognize in schizophrenia as well as many neuropsychiatric disorders. Interestingly, if we put water on the floor and hide those irrelevant stimuli and give the animal the drug, they're no longer impaired. So how do we think about this kind of set of observations? The animals are cognitively impaired, in particular when they have to ignore irrelevant stimuli, but their place cells in their hippocampus seem to be normal. Well, if you look in time, and you look at the very same data as I showed you in the place field plots, but you look at how the cells discharge, one with respect to the other, their covariance, if you will, the PCP makes a massive difference. Here, what's plotted is the activity vectors. I'm going to leave the cursor alone. What's plotted there on the x-axis against the y-axis is time against time. On the diagonal is the correlation of one moment with another moment, with the same moment, and so the correlation is perfect. It's 1. And you can see for the first 30 minutes, the activity is very self-similar. Then it changes. That's when we injected the PCP. The activity under PCP in the middle block there is also very self-similar, just very different from the initial patterns that you see. And as the PCP wears off, the patterns start to switch back to normal, then back to the PCP pattern, and back to normal. You can see that around, say, 70 or so minutes. What's actually remarkable is the PCP pattern. If you look here at about, I would say, 15 or so minutes, there's a stripe there. The PCP pattern actually was expressed when the animal didn't have PCP. It was just a very rare pattern. And the PCP seems to not be inducing a particular novel pattern, but making a latent pattern more common. So, trying to understand these data, we wondered how to think about how neurons code information in general. And so here are two extreme examples. You can imagine neurons coding information very much like a traffic light. Those place cell firing fields, each cell responding to a particular location, and you could interpret that activity to imply that the cell is independently signaling a particular location, and you could take an average over those locations if you want. If you want to signal a different kind of signal, you allocate a different neuron to that. On the other extreme, you could look here at a stadium jumbotron, where no particular light analogous to the cells, no particular light is important for this message. What's important is the co-firing, if you will, or the pattern of activity of lights that are on and off. What's interesting about this that I'm going to show you, if I can control the... Let's try that. Will that work? Love to play. I'm going to skip playing that. I'll show it to you somewhat later.
You were right there. Okay.
There we go. So you can also recognize this pattern. As the cells or as the lights move, as the pattern changes, the pattern doesn't even have to appear all at the same time, and it's nonetheless recognizable. And so we start thinking in recent times about the hippocampal activity from this type of point of view, and I'll show you that it's actually relatively easy to recognize these messages in these unstable patterns. And by analogy, you can imagine an object like this, a cylinder, when projected from one point of view or viewed from one point of view, here by analogy to light sources, it can look like a square or a rectangle. It can look also like a circle. And there are various kinds of transforms that you can use for this. A standard nonlinear dimensionality reduction algorithm we'll use in a moment, called the isomap algorithm, is able to recognize that something like a Swiss roll like this is actually a plane. And
2 minutes.
Thank you. And so if we apply the same transform or the dimensionality reduction, for example, to this movie, get it to play.
Down left. There it is.
There we go. So we can actually treat this movie as a activity vector from recording, and you can see that when projected into this space, it actually is very recognizable. They're distinct game over states, if you will, and they tend to be very stereotyped. So we can do the very same analysis with the activity that we record. In this case, we can now record the activity. It's in mice while the animals do this task. I'll just spare you the details, but if you try to recognize differences in the activity while the animal has not been trained versus after it's been trained in this place avoidance task, we cannot find any differences in their tuning. However, if you look at the same activity vectors plotted in this space, they're very, very distinguishable. And they're distinguishable because of the co-firing relationships. In fact, on the right side, you can see that the distinction has been sufficiently reduced or significantly reduced, and that's because the thing that turns out to be discriminating in the different activity patterns is not where the cells fire, but which cells tend not to fire with other cells. When we remove those anti-co-firing cells, this is the kind of pattern that we get. So I'll just wrap this up. In order to test this idea that the anti-co-firing cells are actually what is controlling the representation, what we did was we used a genetic trick to tag the neurons that were crucial for the animal remembering this task. And I won't spare you. I won't tell you the details of how we do that, but suffice to say that we can do that, and we can image them with looking for yellow fluorescent protein, or we can control them with light, channel rhodopsin. And when we stimulate these cells in a completely neutral environment, if I can get this movie to play.
we'll have to wrap it up, please. Thanks. Thank you.
Suffice it to say, those neurons are sufficient for this activity, for the animals to actually express this place avoidance in a completely novel environment. And when we actually look at that activity, it turns out that the stimulation is not making a new pattern of activity, it's simply rotating the pattern of activity in this nonlinear space. So with that, I'll wrap it up. We don't think about schizophrenia in particular, but we think about thought disorder and how thoughts are organized. And we propose the discoordination hypothesis, where the neurons, because of the myriad genetic defects, as you can imagine, are actually fairly normal in their function as individual cells, but the crucial differences that we find is in how they interact with other cells. And there's a mathematical framework for actually making those analyses. So with that, thank you for your attention.
Thank you very much, Dr. Pennington.